In this video, I'm going to explain genetic diagrams and teach you how to construct monohybrid crosses. So genetic diagrams are used to work out what the genotype and hence the phenotype of offspring would be when certain individuals are crossed. So in the genotype, the monohybrid inheritance, where alleles are dominant and recessive, there are three possible genotypes. We could have homozygous dominant, homozygous meaning the same alleles are present and both of them are the dominant alleles. In these examples, you'll notice I've used capital P's of a different font, just so that you can clearly see when it's a capital P and a small p. The second type is homozygous recessive. There are two small p's, and this is the only type at the time that the recessive allele will show up in the phenotype, because there is no dominant allele. Any other time, if there's a dominant allele, the recessive allele will not show up in the phenotype. And the last one, heterozygous, meaning different alleles are present on the genotype. Obviously, because there's a dominant allele, the dominant phenotype will show up. So remembering, phenotype is the expression of those genes, what we actually see in the characteristics. Homozygous dominant, if we go with the example that we used in the previous video, capital P being purple flowers, homozygous dominant will be purple flowers, homozygous recessive will be white flowers, and heterozygous, because it has the dominant allele, will be purple flowers. So let's get started with a genetic cross. The first step is to fill in the parental phenotype, purple flowers, for the female and white for the male. That's just what I've chosen. We're going to use the cross between the homozygous dominant female and a homozygous recessive male as an example to demonstrate how to work out a genetic cross. So we start with a sort of table format, and there we've got the female purple flowers, the male white flowers. As we've chosen to do the example between homozygous dominant and homozygous recessive individuals, the genotypes then are capital P, and small p, small p. So we add those in the next level on our table. The next step is then to realize that those are alleles on the parent's genes. But in order for the parents to reproduce, their cells must undergo meiosis, which means reduction, division so that they have only one set of chromosomes in the gametes. So the formation of gametes is called meiosis, and the cells divide into haploid cells. So in the female, when those two alleles end up in different egg cells or ovule cells, you'll have capital P in one ovule and capital P in the other ovule. In the male, when those two alleles that are in the normal cells divide by meiosis, you'll end up with small p and small p in the pollen cells. So now you know the gametes, and now we can begin to do the cross. So the cross can then be worked out. There's a possibility that each of the two male gametes may fertilize either of the two female gametes. So there are four possible combinations of the genotype, of the offspring genotype. So if we look here, we could have this first female gamete being fertilized by the first male gamete, producing heterozygous offspring, capital P, small p. Then we could cross the second female gamete with the first male gamete, same result, capital P, small p. Then the first female gamete with the second male gamete, capital P, small p. And lastly, the second female gamete with the second male gamete, capital P, small p. So we actually see that in this cross, the offspring genotype are actually all the same. So the next step is, once we've worked out the genotype, doing the genetic cross, we'll be able to work out the phenotype, knowing which alleles dominant and recessive. So for all of those, the genotypes are exactly the same. They all have capital P, small p, which is a heterozygous genotype, and so the offspring, the phenotype offspring, would be purple flowers for all of them. And as a result, we say the genotypic ratio is 100% heterozygous. We wouldn't have any different looking flowers from this cross. They would all be purple. The offspring phenotype is also 100% purple flowers. Okay, so genetic diagrams can also be worked out using a Punnett square. This is a different method. The first method is a full genetic cross. This is a quick way of doing it by putting the alleles on each side of a cube, separating them out. You can see there the gametes are separated out. The female capital P, the male small p, and when we do the cross, P with P, we'll see that all four possible offspring are capital P, small p. And so the genotypic ratio of the offspring is 100% PP, capital P, small p, and the phenotypic ratio is 100% purple flowers, as we've just done with the cross. It's just a quick way, but you may be asked to do either way in your exams. This is called a Punnett square. The last thing that we're going to talk about is pedigree diagrams. This is a kind of family tree, but a family tree that enables us to see inherited diseases and how diseases are inherited. It enables doctors to predict whether a certain couple's offspring might have a genetic disorder or disease. So they used to show how a genetic condition is inherited in a family and may be used to advise individuals whether they're carriers. In this diagram, you can see that the female is affected, the male is not affected. And so when they are crossed, they give rise to 4 offspring, 2 girls and 2 boys, that all are symptomless. But symptomless means that they carry the allele, the recessive allele for the condition. And so they potentially could pass on this allele to their offspring. As you see, the firstborn child on the left here gets married and has children of her own, and one of them is affected. 2, a boy and a girl, are carriers, and the male is unaffected. So genetic disorders can be recessive or dominant. This example here is a recessive allele for a genetic disorder. And we can gather various information from a pedigree chart and use it to study the disease and predict the likelihood of future offspring inheriting a disease. The example here is a recessive allele. The female is homozygous recessive. In other words, that original female at the top of the pedigree diagram, she has got 2 recessive alleles for the disorder. So she has this genetic disorder. And we can deduce this by the fact that the mother is affected, but no one in the F1 generation, the second generation, is affected, but they all carry the allele for the disease. So I will be sending you an example of a pedigree diagram for you to work out, and hopefully this will help you understand this better.